a cardiovascular checkpoint immune checkpoints in cardiovascular disease. And we will have two excellent speakers, and Christian will introduce the first one.
Thanks, Tom. As the chair of the Basic Science Council co-hosting this, it's a particular pleasure for me to actually have two outstanding individuals who had a present and or past affiliation with my institute. And first, I would like to introduce Esther Lutgens, obtained her PhD at the CARIM in Maastricht, and also as an MD specialized in vascular pathology. She then became very fast a professor, affiliated also with the institute in Aachen back then. And when she moved to become a professor in Amsterdam, she joined the affiliation here of our institute in Munich, and kept performing really seminal work on co-stimulatory molecules. She's an outstanding individual, also a double mom, I have to say. And recently, for those who don't know it, she's basically given up on her affiliation in Amsterdam, moved her European operations to Munich, and joined her husband in moving to the Mayo Clinic in Rochester, which is a very big move. And so she's still having two affiliations, but this time in Mayo and in Munich. And Esther, so we very much look forward to learning the most innovative and new excellent insights you've obtained for immune checkpoints.
Okay, thank you very much, Christian, for the nice introduction. Thank you all for inviting me. Let me check. That's my... Here's my slide. Can you see them? So as Christian already told you, I'm going to talk about immune checkpoints in cardiovascular disease. And check. Yeah. The laser pointer. So I thought I would start off a little lightly with some history of atherosclerosis, and then I'll come to the immune checkpoints. So in the 1850s till 1980s, so for a very long time, atherosclerosis was just considered to be lipid accumulation in the arterial wall, just a lipid blob. And early 1970s, there was this response to injury hypothesis that actually the endothelium was injured by oxidative stress. You would get some smooth muscle cell proliferation. Also, cholesterol was still important. And the first immune cells appeared. And people really thought, well, they're just there. They're just doing nothing. They're just sitting there. But since the 1990s, the concept that atherosclerosis is actually an immune disease has really taken off. So now we are at the stage that we consider atherosclerosis a lipid-driven immune disease. And we, of course, still have the cholesterol, the LDL, VLDL, and LP little a, but it's all intermingled with a lot of inflammation. And people really seem to appreciate and started to appreciate that atherosclerosis is actually a lipid-driven disease. Also in terms of therapy, so the immunotherapy has really taken off in all kinds of diseases, rheumatoid arthritis, the cancer field, but also since 2017, we know that if you apply immunotherapy to cardiovascular disease, it's really working. So this is a proof of concept trial, the CANTOS trial, and it's actually a very big trial where they gave canakinumab, an anti-IL-1 beta antibody, and what Paul Whitker and Peter Libby saw together with Novartis is that if you give anti-IL-1 beta, you can reduce the primary and secondary cardiovascular disease endpoints, but you still cannot overcome the increased overall mortality. And there was a little bit increased risk of infection. So I think the field has really moved on, and we think we can develop better immunotherapies than anti-IL-1 beta, which is already quite good, but I think we always can do better, and we should aim for doing better. And one of the targets that could be very interesting is the immune checkpoint. So eventually we really want to reduce cardiovascular disease, but also cardiovascular disease mortality. And we would like to not have any immune suppressive side effects. So that's the big dream. So what are immune checkpoints? Well, if you open your immunology textbook and look at co-stimulation and co-inhibition, you will most likely find this graph. And it's quite old-fashioned, I must say, but the general concept was, and also still is, that if you have a T cell that interacts with an antigen-presenting cell, you need two signals in order to make it proliferate and activate, or on the other hand, inhibit. So you need the TCR-MHC2 interaction, so the antigen-specific interaction, but you also need a co-stimulatory response. And this co-stimulatory response is mandatory to obtain proper immunity. And then you get your cytokines that actually regulate this immunity. However, nowadays we know that these immune checkpoints are present on all kinds of cells and that they are actually mere communicators of the immune system. You have tons of immune checkpoints and you can divide them into co-stimulatory molecules, co-inhibitory molecules, and there's an ever-growing class of other immune checkpoints. So, as I told you, immune checkpoints are present on more cells than just APCs and T cells. So, together with Claudia and Laura, and we involved Claudia Monaco with it, we designed the immune checkpoint atlas of atherosclerosis. We're actually just in the middle of it. We received the first results from Claudia, who did a lot of cytoph analysis with immune checkpoints. And here you can actually see that that there are a lot of data, that there are a lot of immune checkpoints, and that their expression is absolutely not limited to the T cell or the antigen-presenting cell, but actually immune checkpoints are present on all kinds of cell types. So I don't want you being bored with this enormous heat map. We also have a lot of RNA signaling data. But I can also tell you that this was a mouse study, and we actually found the same expression profile in human atherosclerosis. So we took the data set from the Dupree article, and what we actually could appreciate is that a lot of the immune checkpoints, as you can see them listed over here, if you do single cell RNA sequencing, a lot of the immune checkpoints are present on multiple cell types. So we're still looking into this huge data set and try to come up with new hypotheses and new concepts. And also we would like to learn which immune checkpoints are really important in atherosclerosis, or do we have to inhibit multiple to prevent atherosclerosis? But one of the main lessons that we learned from these analyses is that co-stimulatory and co-inhibitory immune checkpoint proteins are increased in advanced atherosclerosis. We know that some immune checkpoints are paired, and most importantly, immune checkpoints switch cell type specific expression patterns during atherogenesis. And this last hypothesis or observation forms the basis of my talk. So, two main questions. Do immune checkpoints exert cell-specific effects? And what are the implications for current and novel immunotherapies of these findings? So I'm reverting back to one of my favorite molecules, that's CD40 and CD40 ligand. And if you look CD40 and CD40 ligand up, and you can see that its co-stimulatory action results in T cell activation and in immunoglobulin production of the B cell and in Ig isotype switching. So this is the classical pattern of CD40 ligand interactions that also many of the people in industry still use for developing their antibodies or inhibitors. CD40 ligand and also its receptor CD40 are important therapeutic targets. They're very profound, very effective, and many companies are currently developing anti-CD40 ligand and anti-CD40 antibodies or inhibitors. And here you can see an overview which diseases are being targeted, and they are deemed safe, and already the first phase 3 trials have started. So it's a very important and potent immunotherapeutic target. What do we know in atherosclerosis? Well, we know already 20 years that if we block CD40 ligand or CD40, that we get a huge reduction in atherosclerosis and that we get plaque fibrosis, loss of inflammation in the plaque, and actually can accomplish a stable atherosclerotic plaque phenotype. We also know that if we block CD40 with a compound that blocks only one part of CD40 signaling, especially CD40 signaling in the macrophage, that we can also reach the same thing. So if we block CD40 only in one cell type, we get also a reduction in atherosclerosis and also a stable atherosclerotic plaque phenotype. So also in atherosclerosis, it can be really controlled. And here we stumbled upon the function of CD40 in the macrophage. And if you block CD40-TREF6 in a macrophage, you can safely inhibit CD40 signaling without compromising the CD40 signaling that's required for T-cell co-stimulation or antibody production and antibody isotype switching. So these are interesting things. So if you block it totally, you'll get this phenotype. If you block a small part of the signaling intermediate, you can get atherosclerosis specific effects without compromising immunity. However, now I'm just talking about totally blocking CD40 or CD40 ligand or only blocking it CD40 in a macrophage. But as you can see here in the Tisney plots, CD40 and CD40 ligand are expressed on many cell types in atherosclerosis. And as a scientist, I always want to know, but why is this? And what does CD40 or CD40 ligand does per cell type, and what does this cell type do in atherosclerosis, and what are the effects, and can we safely block CD40 or CD40 ligand completely? So we entered on a long journey several years ago, and I did this together with Dorothee Otzler and Norbert Gerdes, and also Christian was heavily involved. We generated the CD40 ligand FLOX-ApoE knockout mice and also the CD40 FLOX-ApoE knockout mice. And now I'm going to overwhelm you with lots of data to actually make the point that CD40 and CD40 ligand have cell-specific effects. So let's start with the T cell and the platelet. So Michael, who is a PhD student in Munich, took on the challenge to block CD40 ligand on the T cells and on the platelet and then investigate atherosclerosis. So if you block CD40 ligand on T cells, then you get very nice effects. And because of time, I won't go into deep mechanism, but I just show you the results. So T-cell CD40 ligand deficiency results in a decrease in atherosclerotic plaque area, a decrease in necrotic core content, thicker fibrous caps, and also less CD3 positive T cells. So these are very nice. So if you would target CD40 ligand on the T cell specifically, you would get a reduction in atherosclerosis. However, CD40 ligand is also very abundantly present on the platelet. But if you block CD40 ligand on the platelet, nothing happens. There is no change in plaque area and no change in plaque composition. However, it does the thrombus or the platelet-associated effects that are already known from literature. It decreased platelet aggregation, you had less stable thrombi, but no effects on atherosclerosis. Only when we applied a model of atherothrombosis, then we saw a reduction in atherothrombosis, but not in normal atherogenesis. So in normal atherogenesis, the T cell CD40 ligand is important and the platelet CD40 ligand not so. So further mechanistical analysis showed that deficiency of T cell CD40 ligands particularly dampens Th1 responses. What about the dendritic cells? So T cells interact with antigen-presenting cells, so also dendritic cells. And if you block CD40 on the dendritic cell, you actually phenocopy the phenotype of the CD40 ligand flux mouse. So no CD40 on the T cell means also decreased plaque area, decreased necrotic core content, and thicker fibrous caps. So to summarize, we now know that the T cell DC CD40 ligand axis is very important in atherosclerosis, and that platelet CD40 ligand only contributes to effects that are generated by atherothrombosis. So these are two important things to know. So what about the macrophage? So the macrophage was tackled by Laura and Annelie, and we had the LIS-M CD40 mice. And these also developed less atherosclerosis, less necrotic cores. However, one important thing was there were no effects on plaque fibrosis. So in contrast to the full knockout, we did not see plaque stabilization. Although we did see a nice polarization towards an M2 phenotype, and we found out that actually CD40 deficiency of the smooth muscle cell is responsible for the increase in plaque fibrosis. So if you block CD40 on the macrophage, you get less plaques, less necrosis, anti-inflammatory macrophages, but no effect on plaque fibrosis. So now comes the most interesting story and the most unexpected results. So what if we would block CD40 on the B cells? And Mirte Rijtje, PhD student in Amsterdam, tackled this problem. And we thought, well, let's just do the B cell, let's get over it. We will see a decrease in atherosclerosis and publish that and say, oh, it's confirmatory. However, nothing was less true. If we block CD40 signaling on B cells, we get an increase in atherosclerosis. So we tried diet, non-diet, we tried males, females, and in every group we saw that if we block CD40 on the B cell, you got increased atherosclerosis. So how come? We saw that B cell CD40 deficiency decreases both IgM and IgG. We had expected the IgG because CD40 is involved in Ig isotype switching, but we also had a decrease in IgM, and it appeared that we had a loss of B1 cells. And there are two kinds of B cells. You have your classic B cells, the B2 cells that are important for antibody production, IgG production, and are also dependent on T-B cell interactions. But you also have the B1 cells. This is a more kind of an innate B cells, and it produces natural antibodies, IgMs, also against LDL. And these natural antibodies are considered protective against atherosclerosis. So our mice had less of these, and we decided to check if this was also the case in humans. So we collaborated with Colleen McNamara, and she did site-of analysis and on her patients. She has patients with high levels of coronary artery disease and low levels of coronary artery disease. And when she puts the B cells through the site-of and looks at CD40 expression, she can actually see that the B1 cells of patients with severe coronary artery disease have low CD40 expression. And it was the only cluster in which CD40 expression on B cells differed between low and high coronary artery disease patients. We also could see by correlating them with IFAS data that B cell CD40 was negatively, B1 cell CD40 was negatively correlated with atheroma burden and positively correlated with plaque fibrosis. So B1 cell CD40 seems to protect atherosclerosis, protect against atherosclerosis. Well, to really prove that, because our CD19-CRE hits all B cells, we specifically transferred CD40 negative B1 cells and would see if these B1 cells really are responsible for the phenotype. And so it is. So if you put normal B1 cells in a CD19-CRE CD40 mouse, you can kind of rescue the phenotype. So we could conclude that B1 cell CD40 is atheroprotective. So what did we find? We actually found that CD40 is really needed for B1 cell maintenance. If you don't have CD40 on your B1 cells, they take up lipids and everything like crazy. Also, their metabolism is really screwed up. They have high glycolysis rates, they have high oxidative phosphorylation rates, and they actually just cannot cope with the stress. And we could see that if we expose them to various stressors, especially the lipid, if you don't have CD40, then the B1 cells just die and they cannot produce a lot of antibody of the protective IgMs anymore. So we found out that CD40 is actually needed for protective B1 B cell response. And if you remove CD40 from these B cells, you actually get an increase in atherosclerosis. Just to summarize it, I have proven the concept that immune checkpoints exhibit cell divergent effects and also divergent effects in signaling, which I haven't shown you today. And I use CD40 ligand as a model. This is actually true for every immune checkpoint. And we're currently planning to do these experiments also for GITR, because we really see a nice switch from the T cell GITR in early athero towards a myeloid expression of GITR in advanced athero. But from the CD40 ligand and CD40 fluxed mice, we learned that CD40 ligand blockage in T cells is protective against atherosclerosis. It doesn't do much in platelets, and that if you block CD40, the receptor on macrophages or dendritic cells, it's protective. If you block it on B cells, you actually get more atherosclerosis. And if you block CD40 on platelets, you don't see anything. So this was a huge effort, and many people have contributed to these studies. And I hope you enjoyed it, and I hope I haven't overwhelmed you with so many data. You can always contact me if you want to know the mechanisms and the specifics of every mouse. But the point I wanted to make is that we still have a lot to learn about immune checkpoints in atherosclerosis. Well, thank you very much, and I'll stop sharing my screen. Yeah.
Thank you very much, Esther. This was a wonderful summary and quite a lot of new data as well, which we enjoyed very much. And the second lecture, and I would like to remind everyone to ask questions through the question and answer section on Zoom. And please ask questions of anything you would like to ask or discuss with both of the speakers. And the second speaker today is Professor Holger Winkels, who since 2021 has been appointed as the Endowed Professorship for Experimental Cardiology of Aging at the Medical Faculty and Clinic 3 for the Internal Medicine in the Heart Center of Cologne, University Hospital. He graduated from biology in Aachen and then obtained doctorate in Ludwig Maximilians University in Munich, and spent some time in the labs in Amsterdam, and then in Klaus Ley lab in La Jolla Institute for Immunology in San Diego. Now he's back in Europe, and we are very happy to hear about his new exciting studies looking at immune aspects and mechanisms of atherosclerosis. Holger, please. Thank you very much for coming.
Yeah, thank you very much, Tom. All right, let's see. Can you see my slides?
Yes.
Okay, perfect. Yeah, thank you very much for inviting me, everyone. And it's a great pleasure to actually present here in the same session with my former mentors, Christian and Esther. So that's already where you have some overlap in costimulatory molecules, but I'm gonna diverge a little bit on the other side of the axis and rather talk about ApoB reactive CD40s. That's something that I've been focusing on during my time in Klaus Ley's lab. So as Esther already nicely introduced, we now appreciate that atherosclerosis is a chronic inflammatory disease, which has also features of an autoinflammatory disease. And what we mean by this is that we find B cells and also T cells that respond to antigens related to atherosclerosis. And one of the key antigens that we find in atherosclerotic plaques is actually LDL. or oxidized LDL, which carries, of course, all the lipids into the lesion, but also has a protein component. And already, yes, in the mid-90s, Göran Hansson and his team identified that there are T cells in human atherosclerotic plaques that respond to particles of ox-LDL or even native LDL, as shown here in this hybridoma culture here. So as Esther already introduced, a naive CD4 T cell needs to recognize cognate antigen, which is for CD4 T cell presented in MHC class II complexes by antigen presenting cells, and it needs to receive proper co-stimulatory molecules. We then think that secondarily these activated CD4 T cells home to the atherosclerotic lesion, and based on their subtype will either lead to the progression of atherosclerosis if they are pro-inflammatory subtypes, or lead to the reduction of atherosclerosis if they obtain a regulatory phenotype and produce anti-inflammatory cytokines. So inspired by all these studies from Hansson and similar work from Hansson and also Nielsen and Pridimancha, we basically also wanted to discover in Klaus's lab whether there are antigen-specific CD4 T cells in patients that would respond to ApoB peptides. And I'm just showing here a little snippet from all the similar work that we had. We first predicted parts of ApoB peptide that would bind to MHC class II complexes. And we particularly in this study focused on one particular sequence of ApoB. It's the P18 sequence that is sequence identical, the only sequence that is sequence identical in ApoB in the mouse and in humans, and it binds MHC class II in mice and humans with a decent affinity. So we used this peptide and in collaboration with Bill Kwok made tetramers, which are artificial MHC class II complexes loaded with the ApoB peptide, linked together via streptavidin and a fluorochrome. So it allows us to detect these antigen-specific cells in patients that have the particular MHC class II complex, in this case, the DRB0701. And when we added transcription factors to this very complex analysis, we saw that in patients or healthy volunteers, we should say, the majority of ApoB reactive CD4 T cells express FOXP3, but there's also some cells that express some transcription factors related to very pro-inflammatory subtypes of CD4 T cells like Tbad or ROGAMA-T. And this landscape kind of changes in patients with subclinical cardiovascular disease. We have an expansion of the ROGAMA-T positive T cells, particularly the ones that are co-expressing FOXP3 and ROGAMA-T. We think that basically atherosclerosis leads to Treg fragility and to the loss of some of their identity. We then followed up later, Dennis Wolf, and at the time in Klaus's lab with mouse studies where we looked deeper into the mechanisms, and he identified that with a different peptide sequence that also CD4 T cells responsive to ApoB have a huge number of ROGAMA-T positive cells that indicate Th17 cells that are very pro-inflammatory. And to a lesser extent, we have Tregs in CD44 activated ApoB cells. That is very different in ApoB negative, so in the bulk population, but the majority of cells is actually of a regulatory phenotype, and the minority expresses Th17 features. When we followed this over time, we see here that the number of Tregs slightly decreases in atherosclerosis, but in the ApoB compartment particularly, it almost drops to 0. And we have the occurrence of Tregs expressing, again, transcription factors from other lineages, such as here ROGAMA-T for the Th17 Treg, again, showing that we have plasticity and regulatory T cells in atherosclerosis. But we also see in the course of atherosclerosis, very pro-inflammatory Th17 subsets, which are not present in the beginning of atherosclerosis. So the question was now, are these fragile Tregs really losing their identity, or do we really have, or do they keep stably expressing FOXP3, or do we have a mixture? What is exactly happening here? And in the first attempt, we made use of a Treg tracker mouse, which co-expresses under the control of the FOXP3 promoter, the Cre recombinase, and a yellow fluorescent protein. We crossed them with a ROSA 26 flox stop flox red fluorescent protein mouse and ApoE knockout mouse. So what basically now happens is that current Tregs will be red and yellow because they co-express YFP and RFP, and if the cell in the course of atherosclerosis would lose FOXP3 expression, it would lose yellow fluorescence and thus become only red fluorescent, which we can detect in flow cytometry. And we have enumerated this here on the right, and if you keep these ApoE knockout Treg tracker mice on a chow diet, you see a mild increase in these X-Tregs, but particularly among the ApoB positive cells on Western diet, we have quite a significant increase in X-Tregs. So we really have not only Treg fragility, so Tregs keeping FOXP3 expression and showing transcription factor expression of other lineages, but we also have loss of regulatory T cells in atherosclerosis. And there have been several reports that Tregs can obtain multiple fates. So Dennis and in the lab showed, and Klaus's lab showed that they can obtain a Th17 phenotype. Work from Elena Garkina and Klaus Ley earlier on showed that there are Th1 Tregs that also have a reduced protective effect, and they express a lot of interferon gamma and pro-inflammatory cytokines. And in Lynn Hadwick's lab back in the day, Dalia Gades showed that actually Tregs can convert into BCL6 mice. The problem is BCL6 cells which indicate T follicular helper cells. One of the problems here is that all the lineage tracker mice used here in our initial lineage tracker mouse are not temporarily precise. So we have a constant output of Tregs in mice. And we were actually designing a new experiment to follow a precise population of Tregs in time and to define in which fate they actually convert. And to do so, we crossed a FOXP3 GFP Cre ERT2 mouse with the known ROSA 26 RFP mouse and the ApoE knockout mouse. So this mouse allows, is having all Tregs green fluorescent and with a Cre activity that is tamoxifen inducible. So what now happens when we treat these mice with tamoxifen is that the Cre recombinase is permitted to enter the nucleus, excises the stop flox cassette, and permits red fluorescence in Tregs specifically. So that means all Tregs that have been tamoxifen induced will be yellow now, and X-Tregs again lose FOXP3 and thus EGFP signaling, and will be only red because the Cre labeling is genetically permanent. So we first designed a kinetic experiment to see how do we actually, does the formation of X-Tregs develop. So at the age of 8 weeks, we induced tamoxifen in these Treg tracker mice, and then we harvested mice 4 weeks, 8 weeks, 12 weeks, and 20 weeks post-induction. And you see here that in time, we have a reduction of the yellow marked cells. That means the tamoxifen-induced Tregs in the lymph nodes, partially in the spleen and also in the aorta, whereas the red cells, which are X-Tregs, are increasing in time in atherosclerosis, saying that after progression increases the number of X-Tregs. And most of these X-Tregs, particularly in time, upregulate CD44 expression, which indicates that they have an effector phenotype. And we further phenotyped these X-Tregs and compared them to Tregs in intracellular cytokine secretion. You see that X-Tregs, as expected, produce less IL-10, more interferon gamma, so it's suggesting that these X-Tregs are more pro-inflammatory and less regulatory, and some even express IL-10 and interferon gamma in both. We also see presence of CXCR5 and PD-1, markers that would indicate T follicular helper cell phenotypes and a reduction in IL-17. So what we actually see here is that X-Tregs share features of T helper 1-like cells and T follicular helper-like cells, but they don't seem to have the Th17-like phenotype, which we have seen previously. We furthermore tested for the suppressive capacity of Tregs and XTregs by sorting them and subjecting them to a classical in vitro Treg suppression assay and measuring as it readouts the proliferation of effector T cells that are stimulated. And you see here that Tregs in a dose-dependent manner nicely suppress proliferation of conventional T cells, whereas XTregs fail already early on to suppress these effector T cells, suggesting again XTregs are rather pro-inflammatory and pro-atherosclerotic than regulatory T cells per se. We further now wanted to look deeper into the cell types and FACS-sorted Tregs, conventional T cells, and XTregs, and subjected them to bulk RNA sequencing. And what you can nicely see here is that each of the three different populations sorted either from lymph node or spleen from multiple mice is clustering among their own identity. So Tregs form their own cluster, conventional T cells do so, but also XTregs do so. And it appears here that XTregs are transcriptomically very different from regulatory T cells and conventional T cells, although we would rather think that XTregs share more features of effector conventional T cells. So just to give you a primer here of the transcriptome analysis, which we're currently still undertaking, you see that XTregs only have 50 genes upregulated in comparison to Tregs, whereas Tregs have more than 500 genes upregulated, or on the vice versa, XTregs have downregulated these genes. And when we did pathway enrichment analysis, you see in these XTregs that they seem to be more active in translation, whereas Tregs have to are enriched for pathways of cell cycle and mitosis. And we're currently still following up of what that exactly means. So one of the hypotheses that we're following is what we were wondering is how do XTregs actually form? What's the mechanism behind it? And one of the ideas in the field is that constitutive TCR triggering and expansion of Tregs will lead to the conversion of these cells. And particularly in atherosclerosis, we have LDL cholesterol all the time around. It's a non-dispensable autoimmune antigen. And so we thought if now XTregs will particularly be triggered by constitutive TCR signaling, we could find a TCR clonality enrichment in the XTregs over the Tregs. So that's what we did here. We sorted Tregs and XTregs and performed TCR sequencing, and we can see that actually the clonality is higher in the XTregs, suggesting that XTregs are more related to each other and have been induced by constitutive TCR triggering. And we further can see that in a way by comparing the TCR repertoire of XTregs and natural derived Tregs that stem from the thymus. When you look at the clonality or let's say individual TCR sequences, which we find in conventional T cells, XTregs and Tregs, conventional T cells and XTregs only share 80 TCR sequences, whereas the overwhelming majority is shared between Tregs and XTregs. These Tregs come from the thymus and are relatively thought to be stable under homeostatic conditions. Conventional T cells also can give rise to XTregs, which are rather unstable. What we think here, though, is happening is it's not a conventional T cell switching to a Treg that loses its expression and turns into an XTreg, but we have the way from natural derived thymic Tregs going into XTregs. And so now what about antigen-specific CD4 T cells? And we first designed here an experiment to actually detect which is the most immunodominant epitope in atherosclerosis in mice. And we needed to do that because XTregs represent a very low frequency among lymph node cells. And we vaccinated here black 6 mice, and you see that the vaccination with the P102 peptide of ApoB100 expands CD4 T cells drastically, and we measured this response here by a tetramer and compared it to adjuvant controls only, or controls that carry a different MHC complex where you would not expect binding. And what you can see here now when we look into atherosclerotic mice is that we have in the CD4 T cell compartment over time a induction of P102 memory CD4 T cells 12 weeks in post-induction, so that means in 20-week-old mice, where we see already atherosclerosis in the spleen and a steady decline in lymph nodes. It seems to be that antigen-specific T cells have a compartmentalization in a secondary lymphoid organ in the course of atherosclerosis. And how does this look now for our X-Tregs and Treg compartment? And here you see the frequency of X-Tregs among the ApoB positive cells, and you see that already 8 weeks post-induction, so at the age of 16 weeks, we have almost a plateau of 20% X-Tregs among this population, and only a steady incline in the spleen, whereas we have a stark decrease in the Tregs and among the ApoB reactive CD4 cells. Just to compare this to the bulk population shown here, the enrichment of X-Tregs is at a higher level than in the bulk population. And as a last snippet, I wanted to show you something that we are currently following up on, where we sorted ApoB reactive CD4 T cells from vaccinated mice in single cell, single well fashion and subjected them to a deep RNA sequencing SMART-seq2 protocol to identify their transcriptome. But what we also can do bioinformatically here is to rederive their TCR sequences. And what was very interesting to us is that among the only 140 cells that we sorted, which are not from X-Treg mice or any mice that have been used in the previous studies, but completely normal black 6 mice, we find 11 clonotypes that are shared with the X-Treg population in atherosclerotic mice. So that kind of suggests that we have already in healthy mice X-Treg or Tregs that are prone to switch into X-Tregs that have public TCRs. And that can be of a bigger implication when we're thinking to use antigen-specific, ApoB-specific CD4 T cells as a vehicle in a vaccination strategy against atherosclerosis. So we need to be, I think, very careful here in how and when we are administering vaccines, because exactly we don't want to enhance pro-inflammatory or X-Treg phenotypes. So with this, I hope I could show and convince you today that ApoB reactive T cells are present in healthy and cardiovascular disease individuals, that X-Tregs with a pro-inflammatory phenotype accumulate in the course of atherosclerosis, and that X-Tregs arise from N-Tregs, likely due to constitutive TCR triggering, but the exact mechanism is so far not well understood. And with this, I would like to thank everyone involved in these in this work. It was mostly driven by Amal and me back in Klaus's work in San Diego, and our collaborators, Dennis Wolf, Bill Kwok, and Mark Jenkins, and all the funding and agency. And I thank you for your attention, and I'd be happy to take any questions.
Thank you very much for this interesting data and stimulating thoughts that not everything that we thought is protective will always remain such. So now we are open for discussion. And again, I encourage everyone to ask questions through Q&A section of in Zoom. Please type your questions and we will discuss. But now I also encourage panelists to ask questions. Maybe I can start with a question coming from our audience. It's directed mainly to Esther, but maybe Holger can comment as well. If we look at the cell specificity, for example, of targeting of CD40 on different cell types, how are we going to sort of use this therapeutically, yeah? Considering that the effects may be different in different cell types.
Yes. I think there still needs to be a lot of research, because also most of the cell types also have slightly different signaling intermediates. So if you would target the signaling intermediate specific to CD40 or CD40 ligand signaling in that cell type, that would be a strategy. But I also think there is a lot of challenges for the pharmacy and the industry to really develop targeted treatments. So you can, for example, with nanomedicine and nanoparticles, you can already beautifully target macrophages. We currently found also that there are specific of formulations that specifically block or target the B1 cells. So there's still a lot to be discovered, and we just know that these molecules exert really cell-specific effects. So the targeting is still under development, and we have CAR T cells. So there is a lot going on in targeting cell type specific actions. You have some bispecific antibodies, etc., but it's still totally in development. So there's a lot to explore and a lot to be tried out.
And I think Magdi El Habal, related to this, is asking if you can clarify your way of targeting specific cells at the time when you inject systemically. Yeah.
The
first question here. Yeah. So I think.
Yeah, so we tried several things. Targeting macrophages are easy. So we did that with our compound. We could specifically target our compound to the macrophage. So that's an easy one. And we can also, using bispecific antibodies, we could target specific cell types. But I haven't gotten the results of that yet, but it's a promising field and we're certainly working on that.
You can see Pasquale.
Has a question, yeah.
Okay, I will do it. So I have a very similar question, Esther, related to the systemic effect of targeting co-stimulation. So what data do you have in terms of what happens in terms of immunosuppression in the short and longer term?
Yeah, so in the of course, depends on how you define short-term and long-term, but if you are treating for about 8 weeks in a delayed setting, after those 8 weeks, if you specifically target, for example, CD40 in the macrophage, there are no signs of immune suppression. There always will be some intervention of the immune system because you target the immune system, but doing this cell type specifically and signaling specifically, you could really limit this immune suppression by just keeping other pathways intact. For example, if you target CD40 TREF6 in a macrophage, you leave CD40 TREF2 signaling untouched, so your B cells will function normally because they highly dependent on CD40 TREF2 and not on CD40 TREF6 signaling. So these are ways you can circumvent the systemic effects. But there's a lot to learn, and yeah, we just learn it step by step, and there will be many more insights in how these immune checkpoints work and exert their effects, and we will also get a lot of insights into their downstream pathways and thereby get the tools to really target these downstream pathways specifically, and thereby we will prevent immune suppressive side effects, but still in the future. So we make small baby steps, but they look very promising.
I have a question following up on that, maybe, because someone was also asking or referring again to the B-cell mediated mechanism, which is obviously an effect also on IgM levels, as you pointed out. And I think there's a remarkable resemblance in the phenotype to the B-cell specific CXCR4 knockout that we and Colleen McNamara have also shown. So I'm wondering if you basically have both of these protective effects actually in B cells. Maybe have you thought about maybe converging those two signaling pathways, and maybe, as you pointed out earlier, maybe you can thereby define the hub that you might want to basically target then in the end.
Yes, so we're working on it and defining the differential signaling pathways of CD40 in a B2 cell and in a B1 cell. And our first data somehow seem to hint that CD40 in a B1 cell can interact with TREF5, but these data are really preliminary, whereas it's in a B2 cell, it's certainly TREF2. So this is one divergent effect, and the other way is targeting the B cells. So you could very easily target B1 cells with HDL nanoparticles. They go in, they won't go into B2 cells. So that's another specific way to kind of specifically target it, because in the B1 cell, you actually want to activate or maintain CD40 signaling in order to be it more stronger and more protective and more capable of producing the natural antibody. So this would be a different way to go.
That would exactly mimic that effect of CXCR4. I was maybe wondering whether you could sort of think about some sort of dual agonism that then would again be B cell subtype specific.
Yeah, that would be very cool to do.
And maybe if I can follow up with another question related to Esther before we get a little bit more to Holger. There's Zuan Xu from China asking because of the role of the immune checkpoint inhibitors also in tumor cell immunity in terms of, you know, how for instance with PD-1 also, if you use that as a target in atherosclerosis, and how far that would also mean to expect side effects in other, you know, immune surveillance systems.
Yeah, so we actually checked this hypothesis. So we did a dual immune checkpoint inhibition, CTLA-4 PD-1 in atherosclerotic mice, and what we could actually see that we got a huge CD8 response, as you would expect. We did also see, we did not see an increase in atherosclerosis, but we saw plaques that were highly inflammatory, and that also explains the cardiovascular symptoms of patients who have that. So the thing is, what we did not test is these tumors are very well cleared by immune checkpoint inhibition. So we're currently developing a model, a combination model, athero and tumor, and we have the first results where we want to apply immune checkpoint inhibition and then see what this dissolvement of the tumor does to the atherogenic response. And there's also something with cell type specificity, because if you look at our immune checkpoint atlas, we can actually see that the more advanced the athero is, the more PD and PD-L1 is being expressed by the macrophage instead of only by the T cell. So there's still some ways to tweak around with that. And in athero, we won't use the immune checkpoint inhibition, but we actually want to activate the inhibitory immune checkpoints. So yeah, those are also ways to do it.
Well, Tom, do you want to continue? I can.
No, I think we also have some questions to Holger, which actually it also came to my mind. Is your transcriptomic analysis, for example, hinting towards the ways that you could prevent this sort of change in phenotype of the Tregs and sort of stabilize them in a protective sort of phenotype?
Yeah, that's a very great question, and I think we have to be very cautious when we are interpreting these results, because at the time we are looking at it, the Tregs are already X-Tregs, and it's very hard, and we have not found out to find this transitory phase of Tregs that are in the progress of switching. I think that's where we would learn the most, and then there are many pathways that can contribute to the formation of X-Tregs, and it's very complicated. So we know that pro-inflammatory cytokines such as IL-6 and interferon gamma in the right context, for example, in a tumor can lead to Treg fragility. But TNF-alpha then again stabilizes Tregs via TNFR2, which is highly expressed on them. And we know that IL-2 has to be available for maintaining a Treg phenotype. Tregs need sometimes a TCR signal, but not constitutively for their maintenance, and metabolism is also has a huge effect. There are conflicting results on hypoxia, which we find in tumors, but also in the atherosclerotic plaque, whereas some reports say HIF-1 alpha stabilizes Tregs and is induced by hypoxia, and in others it says it destabilizes Tregs. So I think that it is still not yet clear why in the course of atherosclerosis we have this switch, but I think it is this constant pro-inflammatory hypercholesterolemic environment that triggers these cells to gradually cave and turn into a dysfunctional Treg.
And in single cell RNA sequencing, can you gain any sort of insight into this progression between different cell types?
Yeah, what we have seen in single-cell RNA sequencing analysis, and we're currently still analyzing from vaccinated mice these single-cell transcriptomes, is that the Tregs seem to convert gradually via a Th17 phenotype towards the Th1 TFH phenotype. And what we have seen now in our study here is that the XTregs convert 20 weeks past, so at the age of 28 weeks, into this Th1 TFH phenotype and don't have so much a Th17 phenotype anymore. So we think it is a gradual progression. But one interesting aspect could also be that there is within the Treg community susceptibility to turn into a particular XTreg phenotype. And it's not clear if XTregs phenotypes can then convert from one into another.
Well, thank you. Maybe I follow up on this, because this is also like a cause and consequence, a hen and egg question. You know, I mean, you showed the inducible model to induce a Tregs, but on the other hand, you know, it's never quite clear whether the loss of FOXP3 could also be in itself a causative effect in this inflammatory clonal conversion. So I was actually wondering whether you could also apply a model of conditionally just or transiently just deleting or depleting FOXP3 alone, you know, without anything else.
There are models where you can reintroduce FOXP3 again in Tregs that have lost FOXP3 expression. We have not done any studies in that regard, but that is very interesting. There is reports out there that you can induce FOXP3 expression again in XTregs, but only in a certain subset. And that is the ones that don't have a, or that still have a partially demethylated FOXP3 promoter region. So if the entire FOXP3 promoter region is methylated, then there is, it's very unlikely that you can induce a Treg phenotype in these cells again. So I think it is, again, a subspecialized population there.
Thank you. I don't know, AJ and Pasquale.
I had a question for our speakers today. Both sets of studies that presented today use Cre recombinase to target their gene of interest and induce their phenotype. The phenotype that are studied are low level of inflammatory reaction. The Cre is notorious for activating interferon response, both alpha and gamma. I wonder how the investigators that presented their data today discern the effect that results from interferon response in response to expression of Cre recombinase from the true effect that they see from manipulating their gene of interest.
So I don't know who can answer first, Esther, maybe, and then Holger.
Yeah, so that's indeed true, but we also use appropriate controls. So we also use the Cre, we use subgroups without the flox mice and the flox mice without the Cre and the Cre and the flox mice. So that kind of separated. And also the Cre recombinase, the interferon response is quite low and less strong than the absence of the gene itself. So I think that has been well corrected for. So
very important to emphasize these methodological aspects of designing such experiments and using appropriate controls. Holger, do you want to add anything?
Yeah, definitely. I mean, we have been using a mouse model, which is or has been published 10 years ago. And there we see already in the early reports that the Cre activity per se is not affecting Treg stability in itself. So under homeostatic conditions, using this mouse model, there's almost no Treg conversion to be seen. So we would argue here that the effect of the Cre induced interferon response is not affecting Tregs very much in our setting. It's more so the homeostatic environment or the that causes this Treg conversion. But it's a great question, and it has to be controlled cautiously.
Thank you. Pasquale? Your microphone.
I'm mute. One quick technical question for Holger. When you see the differential effect in the spleen versus the lymph node, which lymph nodes are you referring to? The draining lymph node like aortic or renal, or the irrelevant lymph nodes not draining the vessel?
I think in this study we have used the mesenteric, the cervical, and the periaortic lymph nodes. And the reason being is when we are feeding a high-fat diet in mice, we see that in the first couple of weeks the mesenteric lymph node is inducing and expanding inside. And that moves on later onto the cervical lymph nodes that are increasing in size when we continuously feed a high-fat diet. So we think that there is a systemic T-cell activation, not only in the draining lymph nodes, but also in remote lymph nodes. And in time, it changes in the body of the mouse. But we don't exactly know yet what that means here in this context. So we just included everything that we have known from previous studies and looked into these lymph nodes.
Okay, thanks.
Thank
you. Christian, do we have any further question? There is a question from Gabriele Trocetti, which I think was not answered yet. It's close to what was addressed before, but it's about predicting different phenotypes in terms of...
Exactly. So I think this is what Esther already referred to when she mentioned the immune checkpoint inhibition in the context of a tumor model and its effects then on atherosclerosis. Here, the additional question would be in how far that would have effects on plaque rupture in these animals, which I think, is hard to predict, but will probably come from the experiments.
So I think...
Other than that, I think we have covered everything.
So thank you very much to both speakers. Maybe Christian, would you like to close?
Yeah, so it was a pleasure to see this basically different views of the immune landscape in atherosclerosis again, and also some new targeting possibilities. And I think we had a very good session, and with a good audience, like Tom referred to. The YouTube streaming is probably even further adding to this. And we expect another interesting webinar in December on cardiovascular remodeling with Eva van Rooij. So we hope to see you back then, and we're really eager to continue this format also, beyond the pandemic or anything that is going to happen.
Thank you.
Thank you.
Thank you.
Yes.
Thank you.
Once again, thank you very much, Esther, and
thank you for inviting. Cool data, Holger. That's nice.
Thank you for inviting, Tom. Yeah, thank you, Esther.
Oh, very nice.
Very nice. We hope to